Okay, could somebody confirm you can hear me?
Yeah.
My computer is going to be a little slow because it's still working on the lecture lesson, lecture movie, whatever. Let me share my screen. Are there any questions? Let me state that I forgot to mention that I did do some grading, and one of the labs for last week is graded. I hope to get the other lab graded tonight. We'll see if we succeed in getting that done. And I've not sent out the unknown numbers. I hope to get that sent out tonight too. Oh, look at that. That lecture Zoom lesson just finished, so that'll speed up my computer. All right, are there any questions about any of that? Let me stop sharing my screen and then I'll take a look at the actual lab. I think it's lab 5 I got graded. Close that down. Yes, lab 5 is graded one time. I'm working on lab 6. Lab 5 must be, if you want to, what do you say? If you want to send in corrections, you have until this Saturday to get that graded. I had lab 5 graded on Tuesday or earlier. On lab 6, I'm not sure. If I get it graded tonight, you may have only until this Saturday to get it corrections. But if I don't get it done tonight, I'll give you another week to get lab 6 done. So I'm not sure when lab 6 will be the final date for putting in corrections will be. Any questions about any of that? All right, let's get out of the grade book. And share my screen. We're a little behind in the lecture and hopefully we'll be able to catch up. In the lab, we're right on schedule. Today, we're going to cover lab 7, throat and urine cultures. And hopefully I'll send out at least your number for your unknown work. There's nothing really for you to do until next Tuesday, when I do hope to send you the results. Any questions about what we're doing? All right, let's begin lab 7. So as is normal, lab 7, throat and urine cultures, you do have some reading to do. So you should read about nose, throat, and mouth normal microbiota, and that's actually only page 392. You should read about the beta hemolytic streptococci, and there's some text at the end of this module for that. You should read about Haemophilus. There's also text at the end of this module discussing that. You should read about the oxidase test, and that's page 132 if you have the 3rd edition of the custom edition textbook made for Clark College. You should read about the cadlase test on page 155. And you should read about the coagulase test, which is also at the end of this lab module. Today, we do have some video clips for you to watch in the lab. No virtual lab. We had one, but it uses Flash, which doesn't work anymore. So your learning objectives, if you can successfully complete the lab, you should be able to explain how to properly collect throat and urine specimens for routine laboratory culture. You should be able to list the common bacterial pathogens isolated from both the throat and from urine cultures. You should know the culture media used to cultivate these pathogens. You should be able to recognize which tests are used to identify throat and urine pathogens, and we'll talk about several tests today. And then lastly, you should understand the principles of how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will briefly talk about the XV factor testing, but I'm not going to expect you to understand or interpret that test. All right. So once a specimen is received by a lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated for a throat and a urine specimen. Below is an overview of the culture media used to cultivate common pathogens isolated from the throat and the urine cultures. So in a throat culture, we only have two types of media which are used to grow throat cultures. We use blood agar most of the time, and occasionally, besides blood agar, we also use chocolate agar. Streptococcus pyogenes is the most common pathogen isolated from the throat, and it will grow on blood agar. In young children and in infants, we also can isolate Haemophilus influenzae, and it doesn't grow on blood agar. It grows on chocolate agar. And so if we have a young patient, we have to grow the throat cultures on both blood agar and chocolate agar. Any question about that? All right. For urine specimens, we have three media that are commonly used. And usually, when we have a urine specimen, we grow it on two different media. One is always blood agar. The advantage of blood agar is obviously it grows fastidious organisms, and then it grows both gram positives and gram negatives. All right, so we will grow a urine culture on blood agar. We also will grow the urine culture on one other agar, and that could either be McConkey's agar or eosine methylene blue, which is EMB. And actually, McConkey's is abbreviated MAC. Okay. The reason why we grow it on two is most of the urine specimens, which are pathomens, meaning something causing a UTI, is gram negative, and that will grow on either McConkey's or EMB, and that's why we grow the urine culture on those, or one of those. Okay. But some of the UTIs are caused by gram positives, and to identify them, you need to grow it in addition on blood agar. So the common bacterial pathogens of a urine specimen include Streptococcus, Enterococcus, Staphylococcus, and then species of Enterobacteriaceae. The number one urinary tract infection is actually E. coli, which is a member of the family Enterobacteriaceae. And then there are also Pseudomonas, which come from a urine specimen. All right, any question about that? Oh, Streptococcus, Enterococcus, and Staphylococcus are gram positive. Enterobacteriaceae and then Pseudomonaceae are all gram negative. All right, so the throat may be looked at, and a sore throat will have usually redness And if there's a bacterial infection, we'll see usually white patches in addition to the redness. The most common bacterial pathogen isolated from a sore throat is Streptococcus pyogenes. And note I stated the most common bacterial isolate. The most common thing that causes a sore throat is actually a cold, and that's not a bacterial isolate. Anyways, redness in the sore throat, that's a viral or tends to be a viral. A bacterial will be red, but there will be white patches, both on the tongue and the white spots on the back of the throat or the tonsils. Streptococcus pyogenes causes strep throat. To detect for Streptococcus pyogenes or causing strep throat, you swab the back of the throat and or the tonsils. You do not touch the sides of the mouth or the tongue. And usually you use the tongue suppressor and a swab and touch the back of the throat. Patients who test positive for Streptococcus pyogenes are placed on antibiotic therapy because it's very difficult for a patient to throw off a Streptococcus pyogenes infection. This bacteria has a capsule which tends to protect it from our immune system. Streptococcus pyogenes is fastidious, and it's sometimes called Group A Streptococcus. I don't use that terminology, and they'll abbreviate that gas. I don't use that terminology either. I always call it, as the species name, Streptococcus pyogenes. Any questions about any of that? All right. So Streptococcus pyogenes is fastidious. It requires growing on a blood agar plate. That is tryptic soy agar supplemented with 5% sheep's red blood. And the blood of the sheep supply the nutrients that Streptococcus pyogenes requires to grow. The plate, once you incubate it, is then examined for small colonies with clearing around them, meaning they have a complete zone of hemolysis, and that we call beta hemolysis. Only 2 biochemical tests are required to identify Streptococcus pyogenes from a beta hemolytic bacteria. And that's the catalase test and the basic trace and susceptibility test. Usually when they do a strep throat and the person has a sore throat, at least when I had this, of course, this was many years ago. I think the last time I had it was in college, which was a long time ago. We'll just state that. But they just streaked it on a blood agar. And if you saw beta hemolysis, they just assumed it's probably Streptococcus pyogenes, and then they put you on antibiotics. The normal throat microbiota, someone who's healthy, has either gamma hemolysis or at worst, alpha hemolysis. Okay, meaning there is no beta hemolysis in the normal throat microbiota. If there is beta hemolysis, and let me blow that up. Still can't see it too well. There you go. That little white center in the center of this clearing is actually the bacterial colony. And that's the way Streptococcus pyogenes is. It's a really small white colony, and it has a large ring of clearing of the blood, and that's what you're seeing here. And there's a few in there, and there's lots of them there. But once you have beta hemolysis, you then run two tests to make sure that it's Streptococcus. The first is the catalase test. Bacteria that have catalase can convert hydrogen peroxide to H2O2 in the presence of catalase into water and oxygen. The oxygen being generated creates bubbles, and you just look for bubbles in the hydrogen peroxide, and that means it's catalase positive. So that would be hydrogen peroxide with the bacteria, and that's catalase negative. This is catalase positive. You can see all the bubbles here. Streptococcus and Enterococcus are catalase negative. Most other bacteria that can grow in the air are catalase positive, like Staphylococcus aureus, which can also cause strep throat. Not strep throat, but a sore throat is what I meant to say. So we do that, and if it's catalase negative, then that's one test that shows that it is Streptococcus pyogenes. The second test, which is needed, is the basic trace and susceptibility test. You take the bacteria that is potentially Streptococcus, meaning it's the beta hemolytic bacteria, streak it over the plate, put on bacitracin disc, meaning this is an antibiotic disc, and if you see a zone of inhibition over a certain size, I think it's 14 millimeters, then we say it is positive for bacitracin, meaning it's sensitive to bacitracin. If it's resistant to bacitracin, it's not Streptococcus pyogenes. Streptococcus pyogenes is sensitive to bacitracin. Any question about that? All right. Another organism which can cause sore throats and is a pathogen in the throat is Haemophilus influenzae, a gram-negative coccobacillus. It is common in ear, eye, and or sinus infections in babies and young children. Haemophilus influenzae is fastidious, but it does not grow on a blood agar plate, at least under normal conditions. And therefore, we grow it on a chocolate agar plate. So chocolate agar is the same as blood agar. It's just you add the agar. When the agar is very hot, when it first came out of the autoclave, that lyses the blood, creating the brown appearance. And because it sort of looks like chocolate, they called it chocolate agar. Blood agar is when you let the agar cool, and then you add the blood when the agar is cool, not room temperature. The agar has to be liquid to add the blood. And you then mix it in and then allow it to cool, and the blood does not get lysed in blood agar. Anyway, Haemophilus influenzae can grow on chocolate agar. It cannot grow alone on blood agar. And let me just isolate. You could grow it on blood agar in one case, and that is what you do is you have your blood agar and you streak Staphylococcus aureus in a single streak running this way, and then you streak the Haemophilus influenzae this way on the blood agar, and you'll note that the Haemophilus influenzae is not growing in any place in this part of the blood agar plate. I'll just blow that up so you can see it better. It is, however, growing in a small region along the Staphylococcus aureus streak. And what's happening is the Staphylococcus aureus are lysing the red blood cells in this region, and the nutrients from the red blood cells diffuse into the media, and then the Haemophilus influenzae can grow in this region of your streaking, but it will not grow outside of this region. And we're not going to talk about that further. You don't need to know about that, but you do need to know about that one. All right. Any questions about the throat cultures? If not, let's talk about the urine cultures. Urine cultures are used to diagnose urinary tract infections. Bacteria can travel to the bladder through the urethra because females have a shorter urethra than males. They are more likely to get a urinary tract infection. And it's just that they have a shorter urethra, so it's easier for the bacteria to travel up the urethra to the bladder. If you remember in a previous lesson, we did mention that the flow of urine actually flushes bacteria out of the urethra. All right, so to get a urine culture, you first clean the genital area, and then you catch the urine midstream, which is called a clean catch midstream. You catch the urine in the middle of its flow to reduce the bacteria that come from the urethra from contaminating the urine. All right. It only reduces it. You still get bacteria from the urethra. And that's the way patients prefer to give a urine specimen. However, you can do other procedures to get a urine specimen. You can put a catheter up the urethra, up to the bladder, shown here. The patients don't generally like that. And then there's even something which is even better, because when you put the catheter through the urethra, you can take the bacteria from the urethra and put it into the bladder. So catheterization is not 100% perfect. It's just better than clean catch midstream urine being voided. And then the best way to do it is actually a suprapubic aspiration, where you stick a needle through the abdomen directly into the bladder and then aspirate fluid directly into the bladder. And that one, if it's done correctly, should have no contamination in it. So any bacteria you get would be bacteria that's inside the bladder. Patients, of course, do not like that procedure at all. So once the specimen is received by the lab, 0.01 milliliters, that's 1 microliter, of the urine is placed on a blood auger plate using a special inoculation loop. I've got a picture. Oh, there it is. Sorry, I just went too far. There it is there. That loop puts 0.001 milliliters of urine on the blood auger plate. You just smear it around. And then you take some urine and then put it on either a McConkey's media or an EMB plate. Any question about any of that? So once again, when we have a urine specimen, we always put it on blood auger, but we also put it on either McConkey's media or eosine methylene blue media. Remember that E. coli, the most common UTI, as well as all other coliforms, these are organisms that grow in the colon, or an Enterobacteriaceae, tend to grow, well, they will grow on McConkey's or EMB. And then Pseudomonaceae will also grow on McConkey's or EMB. Interpretation of a positive urine culture is multifaceted. It depends on how the urine was collected, like the suprapubic aspiration. That has no contamination. So if you get bacteria, you probably have a urinary tract infection. However, if you don't use that procedure, you consider what you have. You consider the gender of the patient. Males tend to have fewer bacteria in the urine than females. You look at the number of colonies per mil in the urine, and then you look at the presence of polymicrobial growth. If there's multiple organisms that are being isolated, it may be that there was a poor collection technique. Then again, if there's only one organism growing, that's probably the organism that is causing the UTI. All right. So then once you get to bacteria growing, it's isolated, and then you run different tests on the bacteria. The first we're going to do is coagulase test. The coagulase test looks at the production of coagulase, which converts fibrinogen into fibrin, which will form a clot. And Staphylococcus aureus has coagulase. Other staphylococcal species like Staphylococcus cephrophyticus, Staphylococcus epidermidis do not produce coagulase, so they will not clot the fibrinogen. What you do is you get blood plasma, and then you add the bacteria. The bacteria produce coagulase. You get a fibrin clot. If you do not have coagulase, you will have the plasma stay liquid. The clot helps the bacteria from phagocytosis, meaning it helps protect the bacteria from phagocytosis. So if the bacteria can produce coagulase, the immune system is going to have a harder time throwing it off. There are two coagulase tests. You can have it on a slide. It's coagulase negative, where there's no clumping. And then you can have coagulase positive, which is clumping on the slide. Generally, when I show you the results, we'll have a tube of plasma, and if it stays liquid, that's coagulase negative. If the plasma clots, that's coagulase positive. All right, any questions about that? So Staphylococcus aureus is coagulase positive. All other members of the staphylococcal family are coagulase negative. The most common bacteria isolated from urine cultures is E. coli, which is in the family of Enterobacteriaceae. This family, all members, can ferment glucose, and they're also oxidase negative. However, there is another gram negative, which can be cause of a urinary tract infection, and it's Pseudomonas aeruginosa. It is gram-negative bacillus, just like the members of Enterobacteriaceae, but it is non-glucose fermenting, and it is oxidase positive. In the oxidase test, we use it to differentiate the two families of Pseudomonaceae and Enterobacteriaceae. If the oxidase test is positive, it'll turn purple, showing the presence of cytochrome C oxidase. And if it doesn't turn purple, stays this color, it would be oxidase negative. Pseudomonaceae is oxidase positive. Enterobacteriaceae is oxidase negative. All right, any question about any of that? So in the lab, you're not physically going to be performing any of these activities. Everything will be done online. However, you are responsible for knowing how to perform the tests that we go through in the lab. We have a little video on the catalase test, so watch it here. We have a little video on the coagulase test, so watch it here. We have a little video on the oxidase test, so watch it here. And then answer the questions in the worksheet of the lab exercise 7. And it does have the clips for the videos on the different tests. So 1, what enzyme converts fibrinogen into fibrin? And then 2, down here, name an organism that tests positive for this enzyme. 2, what enzyme detects the presence of cytochrome C in the electron transport system? And then name 2 organisms that test positive for this enzyme. And I can tell you Pseudomonas aeruginosa is one. You can come up with a second. 3, what enzyme hydrolyzes hydrogen peroxide into the products water and oxygen? And then name 2 organisms that test negative for this enzyme. 4, what rapid test is used to differentiate between the following? So A, Staphylococcus and Streptococcus, name the test here. And then B, Staphylococcus aureus from other staphylococcal species, name the test here. And then what test is used to differentiate Enterobacteriaceae from the Pseudomonas family, name the test here. 5, concerning specimen collection for a routine throat culture, what areas of the throat should be swabbed? And B, what areas of the mouth should be avoided? And then 6, what is the most common cause of bacterial pharyngitis? That's inflammation of the throat, so a sore throat. You need to mention the bacterial cause. 7, what test is used by a microbiologist to differentiate between beta hemolytic streptococcus, meaning Streptococcus pyogenes, from other beta hemolytic streptococcal species isolated from the throat? 8, answer the following concerning a child's throat culture. State 2 non-selective agar plates that are inoculated if the child has a sore throat. So 1 here and 1 there. And 2, what organism can be isolated on CAP, but not isolated on BAP? 8C, you do not need to answer. That's why I have it crossed out. 9, list 3 methods that are used to collect urine for routine laboratory culture. 10, list 3 reasons why urine specimens should be rejected for a culture. 11, how much urine is placed on the surface of a BAP agar plate? You have to tell me the amount in either milliliters or microliters. 12, what is the most common bacteria isolated from patients with cystitis? That's a urinary tract infection. 13, identify the Gram reaction. What is the Gram stain of A, B, C, D, and E? And also give the cell morphology, the shape, not the arrangement of these bacteria. If you only give 1, like the Gram stain, you will get half of this question wrong, and that would be, I don't know, 2, 4, 6, 8. That'd be 1 point for a total. Also, if you only give the shape, you'll be half wrong. So you need to give both the gram stain and the shape. And then at the bottom here, let me see if I can get to that. We have some of the area for reading, like a little bit about streptococcus, gram-positive bacteria, a little bit about beta hemolytic streptococci, and then a little bit about Haemophilus. All right. Oh, there's a little bit about the coagulase test down here too. All right. Any questions about any of this? If not, I'll be here until 8 o'clock. The last student leaves to answer questions. So work on